then we'll look at putting B cells as a component of that. So in your lecture notes, we are on page 68. Now, T cells and B cells have their receptors that we talked about, okay, that are recognizing the antigen, and we've defined the antigen. So this image that I said is what we were following, where we have our antigen presenting cell activating a T helper cell, which activates a CD8 T killer or a B lymphocyte. Inherent in all of that is recognition that the antigen is foreign, okay, and not belonging to self. So there are two methods by which antigens are recognized. T cells have one method, antigen presenting cells have another because of the processing. Every cell in your body that has a nucleus, meaning not the red blood cells, because they used to, but they don't anymore. Every cell in your body that has a nucleus has on its surface these molecules that are called MHC1 molecules. If you were in micro, you may have heard of MHC or you may have heard of HLA. HLA is just a form of MHC, another acronym. MHC stands for major histo, which means tissue compatibility complex. All right? There are two classes. There is class 1 and class 2. Obviously, there are two classes. Class 1, notice how this molecule is holding an antigen. Class 1 presents molecules known as endogenous peptides. They're usually peptides anyway. Class 2, exogenous. This is kind of like intrinsic and extrinsic, okay, within and without. So why don't T cells in your body attack your own tissues? They have these molecules that put on your cells proteins that the T cells, as they mature in the thymus, go through a selection process so that those that remain don't interact. They don't have the T cell receptors for proteins that you make yourself. So how does this occur? I'm going to go through this diagram and then I'm going to draw it on the board. So here is a cell. Here is the plasma membrane of the cell. And here is the endoplasmic reticulum. Now we have two sites, two locations within the cell at which proteins are produced. Remember, a DNA gene is copied by messenger RNA. The messenger RNA moves out into the cytoplasm and it interacts with the ribosome. And transfer RNA brings in amino acids and they're linked together and we have a protein produced that functions within the cytoplasm. That's one way, free ribosomes, okay? If that protein is going to be sent to the mitochondria or to a lysosome, or maybe like an antibody is going to be secreted from the cell, then the ribosome with its messenger RNA attaches to the endoplasmic reticulum, and as that new protein is being synthesized, it's inserted into the endoplasmic reticulum. And then a vesicle of the ER will pinch off and go to the Golgi, where it's further modified, and then another vesicle will pinch off and take it to the mitochondria or take it to the cell surface or whatever, okay? So we have two sites, the cytoplasmic protein or the protein surrounded by a membrane. Both of these proteins are endogenous proteins. They are proteins made by the cell, all right? Fragments of these are placed on MHC class 1 molecules and inserted into the surface of the cell to mark the cell as belonging to your body. They're what I call native proteins. They're proteins that your cell made based on its own DNA, all right? So, here is our cell, and here is the nucleus, right? The nucleus puts out messenger RNA, and that can go to a ribosome, and new proteins can be made that are found within the cytoplasm of the cell itself, all right? Or that might be attached to the ribosomal ER, and the new protein is inserted into the ER. Okay, that's what I'm talking about, those two different methods. All right, now, as part of that endoplasmic reticulum, I'm going to just kind of draw it a little larger here, wrong color. As part of that endoplasmic reticulum, it is going to contain within the membrane a molecule which is identified as MHC class 1. Fragments of this protein that are being produced will bind to MHC class 1, and then when it goes to the Golgi, this will eventually pinch off and will form a vesicle that fuses with the membrane of the cell. And now then is inserted into the membrane of the cell out on its surface, containing fragments of proteins made by the cell, and these are endogenous proteins. Okay? Now what about these proteins that are made in the cytoplasm? Everybody with me so far? Okay. Now we have proteins that aren't made and inserted into the ER. They're enzymes or proteins that are made in the cytoplasm that function out in the cytoplasm, like govern glycolysis reactions. These can pass through lysosomal-like molecules, called proteasomes, and that's not something I'm going to test you on, but they're similar to lysosomes. And these molecules are broken down into smaller pieces, and they pass through into the ER through a transport antigen protein. That's for free. You don't have to know the word TAP. I'm just kind of showing you a pathway here. And these little pieces, actually I should make them purple, but anyway, these little pieces pass into the ER, and they can also be inserted onto a major histocompatibility type 1. They are also considered to be endogenous proteins. So whether they are made in the ER, inserted into the ER, or found free in the cytoplasm, the cell is putting it out on its surface. So the key to MHC1 is that it is a protein synthesized based on the genes of the cell. All right? So it is a marker protein for that cell, for your body. Now, what ramifications do we have then if we were to talk about a viral infected cell? Let's say that a virus got into this cell and it caused the nucleus to use the viral DNA to make new viral proteins to assimilate new viruses. The cell is going to treat that as if it was a native protein, right? An endogenous peptide, because it's made under the direction of the nucleus. It doesn't know the difference. And so those viral proteins will also be placed on MHC1 molecules because the cell made them. Okay? What about a tumor cell whose DNA is mutated? Due to that mutation, it could also make proteins it never made before. But now it's making these new proteins that the body doesn't recognize because it was never made before, and yet the cell is making it based on that mutated DNA. So it also is going to be attached to MHC1 molecules and put out on the surface of the cell. To the body, to the T cells, it's foreign, even though the cell made it, because it doesn't recognize it as being produced by your body prior to viral infection or cancer mutation. Now let's say that I have a heart transplant, and the MHC1 molecules on those heart cells don't match my other cells, and so my body also recognizes them as foreign. Okay? So MHC1 molecules, I want you to associate as with endogenous peptides. And this is how they get on the MHC1 molecule. All right. T cells. Every T cell receptor interacts with MHC1 molecules. So I'll add that to this. T cell recognition. Now, before we move on to how T cells work, let's look at class 2 molecules. MHC class 2. I'm going to erase the board. That's okay. All right. MHC class 2 molecules are only found on antigen-presenting cells. Those are the macrophages, the B lymphocytes, and the dendritic cells. Okay. They can bind to T helper cells. Not to T killer cells. They are produced, these are exogenous proteins. What does that mean?
Made outside the cell.
They're made outside the cell. They're non-native proteins. All right? So here we have, say, a macrophage or a dendritic cell internalizing a virus or a bacterium. And then that vesicle containing the bacterium fuses with a lysosome, which contains proteases that break down proteins. And then that fuses with a vesicle that is pinched off of the ER that has class 2 molecules. Only APC cells have the genes to produce class 2 molecules, all right? While they're in the ER, they're blocked. Otherwise, they would be picking up endogenous proteins, okay? So they have this block that keeps them from picking up endogenous proteins until they get to the lysosome, and probably the pH causes this block to come off. So now we have a cell like a macrophage internalizing a virus or a bacterium. That vesicle inside the cell fuses with the lysosome. And the enzymes of the lysosome break this down into smaller pieces. Now that vesicle with smaller pieces of our bacteria or virus fuses with another vesicle which contains our MHC class 2 molecule. And it will bind this exogenous peptide, one of the pieces of the bacteria or virus, okay, that was ingested. That now goes to the surface and inserts this MHC class 2 molecule into the outer plasma membrane of the cell. So only on macrophages or B cells or dendritic cells do we see this appearance, because the other cells aren't phagocytic. Your muscle cells aren't phagocytic, all right? Your liver cells aren't phagocytic. So only these phagocytic cells are going to have the exogenous peptide presented on the MHC class 2 molecule. Now, so this is our APC cell. Dendritic cell, macrophage, or whatever. However, it is possible, can I erase this right here? I need to make my cell bigger. It is also possible for this macrophage to take this same exogenous peptide presented on its surface on an MHC1 molecule.
Wait, can you repeat that?
So an APC cell can present this exogenous peptide on an MHC2 through the method I showed you. It can also present it on an MHC1. That should be impossible. What kind of peptides does MHC1 take? Endogenous. The cell has to make it. Okay, well, it's not making this protein. It's a piece of bacteria that invaded the cell and is being broken down. So how does this occur? I'm not sure of all the steps, but while it's being broken down, some of these pieces of the exogenous bacterial wall escape. And that's why I don't understand how, because they're too big to go through the membrane, so there must be some kind of a protein channel it gets through. But they escape out into the cytoplasm. Now, they're no longer in the lysosome. They are a protein in the cytoplasm. And how do proteins normally get into the cytoplasm? The cell makes it with messenger RNA and free ribosomes. The cell doesn't have a brain. If a cell is in the cytoplasm, it acts as if the cell was made based on genetic information from the nucleus. So this cell goes through a proteasome, which is really just like a lysosome, and gets broken down into smaller pieces. But now these smaller pieces don't go to the MHC2. They do the same thing as if they were exogenous and endogenous. They go through those channels, that tap channel, and they go into the ER, and they get treated as if they were a normal cytoplasmic protein. The cell doesn't know the difference. And so they will bind to an MHC1 molecule and be inserted on the cell as if they were made by the cell. Okay? So it kind of tricks the cell. So I found a short little cartoon animation that shows this. It uses a dendritic cell instead of a macrophage. All right, so here's the dendritic cell, and it can be just as well via macrophage. Here's the virus, okay, and this is the lysosome. So it's undergoing phagocytosis. That's what the APC cells do. It's then going to fuse with the lysosome. I'll put a link to, or at least the address of this up on D2L. And it starts to get broken down. That's the normal process for getting peptides to put on MHC2, right? It fuses with the lysosome and gets broken down. It's exogenous. But now, some of those products that are produced by the lysosomal enzymes leak out of the lysosome. Now they're in the cytoplasm, and the cell acts as if it is an endogenous peptide, breaks them down into smaller pieces, and those go into the ER through the TAP proteins just as if they were proteins made with free ribosomes. And bind to the MHC1. Now they show these MHC1 molecules as free, but they're actually inserted into the plasma membrane. Okay, otherwise when they're delivered to the surface, they would just leave the cell. Get processed by the Golgi. Inserted into the membrane as an endogenous antigen attached to an MHC1 molecule. Okay? So it really is a trick. It's not truly a native protein. It's not even a protein that the cell made, but because it entered into the cytoplasm, the cell treats it that way. So this is why antigen-presenting cells can have both the same antigen on both MHC1 molecules and MHC2. And that brings us to the next step. All right, so before we talk further about T cells and their activation, I want to take a step back. Remember, we did not talk about the thymus when we looked at organs on Tuesday. So let's take a moment here just to look at the thymus, because this is where the T cells mature. Before we look at their activity, I want to take a look at that function. So thymus is back on page 54. No, the bottom of page 54. So here's a small view of the thymus, and you'll notice it looks like what are follicles, lymphatic follicles. Each lobule, the thymus has several lobes, and within each lobe we have smaller lobules. Notice how each one is surrounded by connective tissue. It's very distinctive. In the lymph node, which is the only other lymphatic organ to have a cortex and a medulla, we only have one cortex that surrounds the entire lymph node. In the middle, we have the medulla, near the opening. Here, each little lobule has a cortex, and the light area is the medulla. Has nothing to do with the germinal center, okay? Because there are no plasma cells. The thymus has T cells, not B cells. The stem cells for T cells in the bone marrow produce the cells that are going to mature into T cells. They leave the bone marrow, enter into the blood, and somehow the ones that end up in the thymus stick. If they end up somewhere else, they die, okay? If they're not mature. From the medulla, they migrate out into the cortex. And as they do so, they undergo a series of changes as they mature. And there's two questions that have to be answered. This is called positive and negative selection. The corpuscles are non-functional. They're just kind of a corkscrew of epithelial cells that lets you know when you look in the microscope that you're looking at the thymus, okay? It's really the only use that they're of. So, T lymphocyte maturation, we're talking about either B cells or T cells. We don't want T cells in our body that will attack our body, all right? But to be functional, a T cell has to recognize the MHC1 complex. So that's the first step. They are exposed to all of these cells passing through the thymus that have MHC1s on their surface, and every body cell that has a nucleus does. If they don't respond to it, if they don't recognize it and bind to it with their TCR, they commit suicide. Okay, they kill themselves. They're allowed to live only if they recognize that MHC complex. Because they have to do that to be able to function. All right? The next step is if they respond strongly, if they respond weakly to MHC1 molecules that have self-antigens, they're allowed to live. If they respond strongly, vigorously, we don't want them. That would cause an autoimmune disease. All right? So this kind of explains this view. So here, the first response is if they recognize the MHC molecule, they're allowed to live. If they don't recognize it, they do apoptosis. And apoptosis is APOPTOSIS, cell suicide. All right? Then, the recognition of the antigen, well, this is actually the MHC and the antigen. So if they recognize the MHC molecule, if they respond weakly to the antigen, okay, they're allowed to live. If they respond strongly, they have to die. If they don't recognize the MHC, they have to die. All right, so basically what we're left with then after maturation are T cells that can interact with MHC complexes, 1 or 2. T cells that do not respond strongly to self-antigens. They will only respond to foreign ones. That's the purpose of the thymus. By the time you are 18 to 20 or earlier, that's considered to have occurred, and T cells are no longer sent to the thymus. It's replaced with adipose tissue. And if you have AIDS and all your T cells are killed off, can't make more. Though some research has shown in mice that there might be a few T cells left, it hasn't been essentially very useful in humans to whatever, if there are any left, for that to occur. But the thymus is where this maturation and elimination of T cells that would attack your body or not be functional occurs. Okay? So now let's see how we activate, this is the last thing we're going to cover, how we activate those T cells. What are they responding to? So, here we go. So we're on the bottom of page 74, well no, page 76. We covered the bottom of page 74. Once you've had a chance to go back and read through this, we'll review this on Thursday and then move into B cells, because the first few steps are exactly the same, and in fact I describe them more thoroughly with B cells. So let's use a virus. We're talking about T killer cells. So I'm going to write a reminder over here for you. T killer cells. I want you to associate them with MHC1, with endogenous peptides, okay, and with the molecule, which won't mean anything to you right now, but we will in just a moment, CD8. We're also going to talk about T helper cells. Those interact with MHC2 class molecules, exogenous peptides, okay, and CD4. Just kind of on the side. So let's say we have a virus infection, okay, and we respond to that by making antibodies that kill the virus. But what about the cells that are infected by the virus and are producing more viruses? Okay. So we'll look at one stage on Thursday, but let's say we're going to look at T killer cells. So let's draw our APC first. All right. First step that we're doing is activating our helper cell, our T helper cell. All right. So in sequence, this is an APC. We're assuming it to be a macrophage, but it could be a dendritic cell or a B lymphocyte. It is going to internalize a virus. Is that specific or non-specific? Non-specific. Okay? As a result of this internalization, the virus containing vesicle associates with the lysosome, starts to be broken down. This is what we drew earlier, right? Bigger. And then those broken pieces are attached to an MHC class what? 2. and are inserted onto the membrane of our macrophage. MHC class 2, exogenous protein. Okay. Some of those viral proteins got free and were inserted into the ER, or passed through into the ER, and were picked up by an MHC class 1. Did the cell make those proteins? No. And again, this would never happen in another cell that wasn't phagocytic. So now we have MHC class 1. Okay? So, is our T helper cell going to recognize the MHC2 or the MHC3? I'm sorry, MHC2 or MHC1?
1.
T helper. 2. So here is a naive or unreacted T helper cell. It has a T cell receptor that just happens to match the specific epitope, if you will, of that antigen. Okay? Another T helper cell would have a different receptor and would match it. Specific immunity because of this. All right? A different T helper cell with a different receptor would match a different epitope. Now remember I talked about the CD4 molecule? Okay, so this is stabilized. This makes a protein that helps to stabilize this, known as CD4. The addition that's made by the T helper cell. The addition of that CD4 helps to activate the T helper cell in the interaction between the T helper cell and our APC, our macrophage. And the macrophage releases chemicals that bind to the T helper cell. And cause it to start making its own proteins. This molecule is called interleukin 1. All right, it's made by the T helper cell too, but I want you to think of it primarily as made by the APCs, by B cells and dendritic cells and macrophages. So interleukin 1 is going to bind to the T helper cell and cause it to be active, cause it to turn on its protein assembly machinery. All right, that's part of our picture. Now we have our MHC class 1. What cell interacts with MHC class 1? The T killer cell. So here we have our T killer cell. It also has a T cell receptor that is specific and only recognizes the same epitope as the T helper cell. It is stabilized by a CD8 molecule. The T helper cell, now that it's been activated by interleukin 1, while it's next to the T killer cell, will release chemicals that activate the T killer cell known as interleukin 2. At least this is a numerical sequence. And the interleukin-2 activates the T killer cell. Interleukin-1 from the macrophage activated the T helper. It made interleukin-2, which activates the T killer cell. Now the T killer cell is active. It makes a molecule known as perforin. Remember how the MAC attacks worked? They opened up the membrane. Perforin works in a similar fashion. It also allows enzymes to enter into the cell, and this results in a direct killing of the cell. But we're not done. This is just activation, okay? Yeah, the macrophage might get killed. Our T helper cell is now active, but not dead. It's not like a bee that once it stings, it, dies. It now is active, and it can activate B cells or other T cells. And we'll talk about the B cells on Thursday. But it can go off and multiply and clone itself and activate other cells as long as they have this same receptor. We'll let it go do its journey, and now we're going to focus on this T killer cell. Because it doesn't die either. Yes?
Does the CD8 molecule travel with it? Or?
Yes.
Okay.
And so it can also produce more. Because every receptor on here is can associate with that same type of an antigen. Yes?
And the interleukin-1 is produced by the macrophage?
By the T helper cell.
Oh.
I'm sorry, the interleukin-1 is the macrophage, or the dendritic cell. Interleukin-2 is primarily the T helper cell. Okay, now there's some overlap, but when you read your book, some can be produced by multiple cells, but I'm limiting it to these particular ones here. So now our T killer cell is off. It's going to multiply, undergo multiple series of mitoses, and produce hundreds of more T cells that look just like it. Some of them will stop cloning and will become memory cells. So 2 years down the road, 5 days down the road, they can meet up with the same viral antigen and start becoming active. But those are the memory cells. We're going to stick with one of these progeny of our original killer cell. Okay? It has the T cell receptor on its surface, just like this one did, that recognizes that same epitope of the antigen. Now let's say that it comes in contact with a different, just a regular body cell that was infected by that virus. So here's a virus-infected cell. The virus is hiding inside it, but it's making new and more viral proteins, and it's reproducing by the thousands and millions every day. What has that virus-infected cell done to this cell? I mean, what has the virus done to this virus-infected cell? Hijacked its DNA. So some of the proteins, endogenous proteins that it's now making are viral proteins. So the MHC1 complexes, not all of them, but some of them on its surface, are going to contain various viral proteins. By chance, they end up matching that particular T cell's receptor. It's already been activated through this process. It doesn't need the T helper cell again. It's already had its machinery turned on. All right? So it comes closer. I'll just make its T cell receptor bigger. It interacts with that. It attaches a CD8 molecule, and it starts firing off perforin, which kills the virus-infected cell. Okay. So it's killing the virus-infected cell not because it recognizes the virus as foreign, but because it interacts with the MHC1 molecule and recognizes that antigen as foreign. Okay. Yes?
So it kills the virus-infected cell. Does it kill the viruses? Because can it just get out of the dead cell and go to other cells?
But the cell will no longer be making viruses.
Oh, okay.
So we, on Thursday, we'll see what antibodies do to the actual viruses. Okay? Antibodies, because when you get a vaccine, that causes antibodies, which prevent viruses from, they'll do things to viruses that we'll talk about on Thursday. Okay? That diagram up there is your extra credit for your next exam. I don't ask you to draw because I hate reading other people's drawings and interpreting them. So I do the drawing, I let you do the labeling. Okay?
You're gonna have that drawn and we have to label it?
So I want you to be able to identify the cell, the CD4, the CD8, the perforin, the interleukin 1, the interleukin 2, the MHC1, the MHC2.
The perforin is released from the
T killer.
T cell?
T killer cell.
T killer cell.
So the macrophage releases interleukin 1, which goes to the T helper cell. The T helper cell releases interleukin 2, which goes to the T killer cell. The T killer cell releases perforin, which goes to any MHC1 virus-infected cell that it matches and interacts with. Okay? All right, that's the end of lecture. So on Thursday, we'll review that first step where the CD4 and CD helper cell are activated, and then we'll look at it how it interacts with B cells rather than T cells.